hello everyone, and welcome to this fantastic webinar. Thank you for joining us. The topic of today is Beyond the Drop, Safety and Biocompatibility Challenge of Ophthalmic Medical Device. This webinar is presented by Neotron, ToxHub, and VitroScreen. I'm Luca Vogesi. I'm in charge of marketing and business development in Neotron, and I'm your host for today's session. If you have any type of questions in the middle of our presentation, feel free to use the question and answer box or the chat box eventually, and we will address this question towards the end of our program. So the speaker of today, as you can see here in the screen, we have my colleagues Valeria Mazzoni, Scientific Project Coordinator from Neotron, Carla Landolfi, European Registered Toxicologist, founder and Chief Executive Officer, Toxicology Risk Assessor, Biocompatibility Expert of ToxHub, and Francesco Carriero, Study Manager, Preclinical Efficacy Studies of VitroScreen. So just another info for you, the agenda of today will be very rich of information for all of you. So we start from ToxHub in terms of regulatory frameworks, packaging material compatibility, and biocompatibility evaluation. VitroScreen with vitro study in a real case of ACH model. And Neutron through the use of techniques that can cover extractable and leachable testing. So a very rich and full of argument today in the session of this webinar. So let me briefly introduce Cotecna. So Cotecna, as you can see here in the slide, is a multinational company based in Geneva that provide TIC, so testing, inspection, and certification services dedicated to the supply chain. We have more than 6,200 employees all around the world in more than 180 different offices and laboratories all around the world. So the main, as you can see, expertise of Cotecna is related to the field of food and pharma, agriculture, metals and minerals, certification, ESG solution, and consumer products. We can have also a very quick overview about Neotron. So Neotron, let me say, celebrate this year 50 years of anniversary of activities. We have developed during these years many different analytical services from food, baby food, supplements, ingredients, cosmetics, pharma, many different fields of work. So now it's time to start the webinar. So I leave the word to Carla Landolfi of ToxHub for her first presentation. Please, Carla, now it's your turn.
Thank you. So let me share the screen. I hope you can see right on the screen. And good morning everyone. I'm very pleased to be here today with our colleagues from Neotron who are kindly hosting us and with the VitroScreen team. It's a real pleasure to talk about a topic that is particularly close to my heart, that are substance-based medical devices, especially those applied to the eye. Today, I'd like to walk you through three key points that are essential for understanding the biocompatibility of ophthalmic medical devices. Ophthalmic medical devices come in many different forms, but today we will focus specifically on substance-based devices applied to the ocular surface. The The A is an extremely complex and sensitive site of application, and its unique anatomy and physiology makes safety evaluation particularly challenging. In this talk, we look at the regulatory framework and the classification under the MDR, and finally, we will address some key safety requirements. Before we begin, let me briefly introduce ToxHub. ToxHub is a toxicology consultancy providing specialized non-clinical expertise and regulatory support across the pharmaceutical, medical device, cosmetic, and chemical sectors. Founded in 2020, we became part of the Sensagen Group in 2022, together with our partner at VitroScreen. We bring solid expertise in product safety and regulatory compliance thanks to our long-standing experience in toxicology risk assessment and biocompatibility gained in the industry. So, before we go into today's topic, it's important to briefly introduce the site of application we are dealing with, the eye. The eye is not a single surface. It's a collection of highly specialized tissues, each with its own sensitivities and its own safety considerations. The eye is one of the most delicate and structurally complex organ in the body, and even very small changes in its environment can lead to irritation, inflammation, and discomfort. The anterior segment, where most ophthalmic preparations are applied and act, includes several structures: the cornea, the conjunctiva, the tear film, the aqueous humor, and the iris, the ciliary body, and the lens. Each of these tissues has different permeability characteristics, different degrees of vascularization, and different protective mechanisms, meaning that each one must be evaluated separately when we assess absorption and safety. For example, the cornea is thin, transparent, and highly innervated, forming a very effective but extremely sensitive barrier. The conjunctiva, instead, is more permeable to and highly vascularized, which makes it a potential route for both local and systemic absorption. Other structures like the aqueous humor, the iris, the lenses contribute to maintain ocular health and can be indirectly affected by substances applied to the eye. Altogether, this complexity means that anything applied to the eye must be gentle, physiologically compatible, and supported by a solid scientific evaluation, because multiple tissues are involved and each requires its own dedicated consideration in the safety assessment. Let's now move into the core topic of the today's presentation, the ophthalmic preparations, in particular the substance-based medical devices designed specifically for use on the eyes. They are typically instilled into the space between the eyelid and the eyeball, which is why they require the highest standard of safety and should be produced under conditions comparable to parental preparations. Traditionally, ophthalmic products were solutions, suspensions, and ointments. Today, the field also includes gel-forming liquids, inserts, and even intravitreal systems. Despite the differences, they all share the same challenge. They must be very gentle, compatible, and backed by a strong biocompatibility and safety assessment. Ophthalmic products are far from simple medical devices. Their formulation are often highly sophisticated, combining multiple functional ingredients and excipients, such as enhancer of viscosity, stabilizers, buffer, preservatives, that must all work together, remaining highly compatible and with the ocular surface. This complexity is further increased by the different packaging formats, multi-dose systems with the preservatives or filters and preservative-free single-dose units, each bringing its own challenges in terms of sterility, stability, compatibility, and biocompatibility. For these reasons, ophthalmic devices require a much deeper level of scientific and regulatory attention than standard formulations. Generally speaking, the MDR places particular attention on substances-based medical devices because these products contain substances intended for medical purposes and are inherently more complex. They appear through the regulation because they require a very specific approach in consideranda, article, and annexes. We must clearly define how they qualify as medical devices, justify a non-pharmacological mechanism of action, apply the correct classification, especially under Rule 21, I will see later, and truly demonstrate their quality and safety. First of all, the mechanism of action. This is absolutely essential because for these products, it must be mechanical, not pharmacological, as for all medical devices. The MDR is very clear. If the primary effect is achieved through pharmacological, immunological, or metabolic means, the product becomes a medicinal product. So, ophthalmic substance-based devices must act through a physical mechanism of action, such as forming, for example, a protective film on the ocular surface. This also means that the formulation has to fully support that mechanical mechanism and must not introduce ingredients with pharmacological activity. That's why the pharmacological assessment we generally do is so important. And to demonstrate the mechanical action, we rely on in vitro assays like film forming or mucoadhesion models. And Francesco will show you an example of this in the next section. The second key point is the classification. With the MDR, the old rules were no longer enough because they didn't properly reflect the invasiveness of the potential or the potential toxicity of products made of substances, especially when applied on mucosal surface like the eyes. For this reason, the MDR introduced specific classification rules for substance-based devices that can be absorbed or dispersed in the body. These rules allow us to match the device class to its real risk profile, especially in terms of systemic exposure. Here we have an overview of all the MDR rules that can apply to substance-based devices, but for ophthalmic products, the key one is Rule 21 because it specifically addresses substances that can be absorbed or dispersed through the eye. Under the MDR, all medical devices based on substances now fall under 221, which means that they can no longer be classified as class 1. The lowest possible class is the 2A, and the final classification depends on the intended side of action and the systemic risk. That is, whether the substance, the device, is absorbed and to what extent. For ophthalmic devices, Rule 21 is particularly important because it help us distinguish products that act only on the ocular surface from those that may penetrate deeper or even reach systemic circulation. And depending on the risk profile, these products may fall into class 2B or 3. So, ultimately, the classification is driven by the intended purpose and how deeply the substance, the device, needs to reach to achieve the intended purpose, which naturally leads us to the key question: how do we demonstrate whether or not a systemic absorption occurs? As told, when we look at the ophthalmic products, one of the most important aspects is understanding how substances can be absorbed through the different barriers of the eye. The eye is, as I told you before, extremely delicate, but also highly protected with the anatomical, physiological, and dynamic barriers that regulate how much of a substance is actually absorbed. The cornea is the main limiting barrier, allowing passage only to very small lipophilic or ionic molecules. In contrast, the conjunctiva and sclera are much more permeable, making them key non-corneal routes for both local and potential systemic absorption. And on top of this, tear turnover, blinking, and nasolacrimal drainage rapidly remove most of what we instill, but can also direct part of it towards systemic exposure. So, in practice, absorption can occur through corneal and non-corneal routes, and evaluating all of them is essential to correctly classify ophthalmic substance-based medical devices under Rule 21. To correctly evaluate absorption and therefore assign the right classification, we need to consider all ocular barrier, as I mentioned before, not just the cornea. As I said, substances applied to the eye may cross the corneal epithelium, but they can also be absorbed to the non-corneal routes, such as the conjunctiva, sclera, and so on. This is why the first and most important step is a strong absorption, distribution, metabolism, and excretion EDME-based assessment. By analyzing the physiochemical properties of every ingredient, molecular weight, lipophilicity, ionization, solubility, charge, we can predict whether a substance is likely to cross the very tight lipophilic corneal barrier, or whether it's more likely to permeate through the more permeable conjunctiva or sclera. EDME is the foundation that tells us how and where absorption may occur. But prediction alone is sometimes not enough. When needed, we complement EDME with experimental permeation studies using models such as epiocular or other reconstructed human corneal epithelia. These systems help us understand whether a substance, typically the functional active, remains on the ocular surface where it's intended to act, or whether it penetrates locally or could potentially reach deeper tissues. And the reverse is also true. Relying only on corneal permeation test is not sufficient without ADME data and a proper chemical physical evaluation. The results remain incomplete and are not adequate for a correct risk assessment or for supporting the appropriate MDR classification. We have direct experience with these. Notified bodies have explicitly asked for and endorsed this integrated approach, confirming that only the combination of ADME chemical characterization and permeation studies provides the level of evidence required under the MDR. Finally, when we talk about safety, the MDR and the ISO standards require a very true assessment of all substances that may come into contact with the eye, both formulation ingredients and potential leachables from the packaging. The regulation places particular emphasis on CMR substances and endocrine disruptors, which is especially critical for ophthalmic products, given the potential absorption of small molecules, as discussed earlier. This means that even very low levels must be identified, evaluated, and scientifically justified. It's not enough to say that they are present below 0.1%. We must demonstrate that they are not released at level that exceed the toxicological threshold, for example, TTC values as for ISO 21726, ensuring exposure remains well below any level of concern. In addition, a toxicological assessment is essential to clearly understand the absorption profile, local tolerability, and most importantly, any potential systemic toxicity of both the formulation and any substance released from the packaging. Even for ophthalmic medical devices, we must take into account systemic acute and repeated dose toxicity as well as genotoxicity. This requirement applies to all device classes and the evaluation must follow ISO 10993-17, ensuring that the patient's exposure always remains below any toxicological level of concern. Before closing, I'd like to highlight the relevance, the importance of the biological evaluation plan, because this is really the foundation of a compliant and scientifically sound assessment. A well-designed BIP prevents unnecessary testing, ensures that the formulation, packaging, and overall risk profile are fully aligned with MDR and ISO expectations, and ultimately guides the entire process toward a complete and defensible biological evaluation report. To conclude, ophthalmic medical devices are among the most delicate and challenging products to assess because their safety depends on precise understanding of ocular anatomy, the main barriers that influence absorption, and the specific MDR framework for substance-based device. A robust evaluation must combine absorption analysis, in vitro permeation data, and the full toxicological assessment covering local tolerability and systemic toxicity. And of course, we must ensure the safety of both the formulation, ingredients, and any potential leachables from the packaging. Finally, and today more than ever, with the new risk-oriented ISO 10993-1, now more closely aligned with the ISO 14971, it's essential to adapt a scientific-based, risk-focused approach to ensure that ophthalmic medical devices are evaluated appropriately, taking into account their specific characteristics, their site of application, and the real exposure of patients. Thank you very much for listening.
Thank you.
Be happy to... Yes, thank you. And now I'm pleased to hand over to Francesco.
Thank you, Carla, very much. It was a brilliant presentation. Absolutely many things to put in our minds and learn every day something new. Thank you very much. So Francesco, is now your turn. So please feel free to share your screen and the floor is yours.
Thank you very much, Luca. Thank you very much, Carla. Okay. Good morning, everyone. My name is Francesco Carriero, and I'm the responsible of preclinical efficacy studies in VitroScreen. Today, I would like to describe the procedure of testing in vitro eye drop-based medical device in order to determine their ability to form films on reconstructed tissues, particularly on reconstructed corneal epithelium. Okay, perfect. Taking a step back, the regulation of medical device has undergone a very important update in recent years. Regulation 2017-745, known as the Medical Device Regulation MDR, replaced two old directives, 93 and 90, entering into full force in May 2021. The aim of new regulation, which is designed to ensure the safety, performance, and transparency of medical devices, was to fill the significant gaps in the old regulatory framework, in line with modernizing the supervision of medical devices, the technological innovations, the globalization and complexity of modern medical device, and last but not the least, to improve patient safety. In order to respond to MDR classification requirements, VitroScreen, a member of the European Union Network of Laboratories for the Validation of Alternative Methods, the UNETVAL, since 2014, and also laboratory certified ISO 13485 since 2023, has developed and internally validated an experimental protocol to assess the film-forming properties of medical devices based on substances. The medical devices tested include solid, semi-solid, and liquid formulations. And it's important to underline that the film-forming test is not applicable to gases and aerosols. The film-forming assay was initially developed on reconstructed human epidermises for the evaluation of topically applied cosmetic cream products. And subsequently, the test has been extended and validated on other tissue models, such as human corneal epithelium, in order to study the mechanism of action of medical devices. The film-forming test is characterized by sensibility, meaning the ability of the test to correctly detect true positive, reproducibility, which means the ability to obtain consistent results when the test is repeated under the same conditions, either within the same lab or across different laboratories. And reliability, meaning the degree to which the test produces accurate and consistent results, aligned with known standard or reference data. These attributes make the test scientifically credible and regulatory acceptable. The test, developed by VitroScreen in collaboration with the Department of Pharmaceutical Science at the University of Milan, which led to a scientific publication in 2017, is based on a quantitative approach that uses caffeine penetration kinetics as a tracer of barrier permeability associated with barrier integrity measurement. The reduction in caffeine passage is used as a performance indicator for MDS medical devices' ability to form a physical barrier. The reconstructed tissues are treated topically with medical devices and subsequently with caffeine at defined concentration. After the caffeine application period, receptor fluids in the basolateral compartment are collected for caffeine quantification by HPLC. Why use the caffeine as a tracer? In the film forming model, caffeine was selected as a tracer due to its low molecular weight, high stability, absence of toxicity, moderate hydrophilicity, and they're able to not ionize at physiological pH. These qualities permit it to overcome the tissue barrier, even in the absence of any damages. The method was implemented on human corneal epithelium to meet the needs of customers and consequently of the needs of notifying bodies in the evaluation of the adhesive capacity of eye drop-based medical device composed of hyaluronic acid at different concentration. In the case study shown, which summarizes several studies conducted during the 2025, are presented the film-forming results of medical device composed of different hyaluronic acid concentration. The negative control is treated with saline solution, while Vaseline is commonly used to treat the positive control thanks to its well-known film-forming activity. Furthermore, studying eye drops, a reference substance composed of 3% of hyaluronic acid is often included in the experimental protocol. This substance, being more suitable to be compared with medical device eye drops, showed good film-forming properties and activity, and also excellent tolerability, excluding any occlusive effect on the corneal epithelium. The results in the graph are presented as percentage of the diffused caffeine compared to the negative control by putting it equal to 100%. Additionally, the results could be expressed as microgram of the diffused caffeine in comparison of the applied dose. In order to contextualize the graph and the results obtained, 100% of the negative control represents the high passage of caffeine in a defined period of time, showing any film-forming effect. Conversely, 4% of Vaseline represents the lowest passage of caffeine in a defined period of time, showing high film-forming efficacy. Based on the results obtained and assuming that our case study provides a summary of eye drops tested in 2025, the increase in film-forming activity appears to be directly related to the concentration of hyaluronic acid, with an increase directly proportional to its concentration. We can therefore conclude that the film-forming effect could be related to the presence of hyaluronic acid inside them. However, the ability of the medical device to create a film-forming effect may not only be related to the presence of hyaluronic acid, but also to the general composition of the eye drops. In summary, the experimental design of standard film-forming test conducted in accordance with ISO 13485 is characterized by a defined product treatment, exposure, followed by a defined caffeine application, a defined period of caffeine application. And at the end, the receptor fluid sampling for HPLC, caffeine quantification. However, the test can be customized according to the sponsor request by modifying the medical device exposure of time, investigating the short or long-lasting film-forming effect, and finally, evaluating the possible persistence of the film after a quick and gentle rinse. In conclusion, why should I perform a film-forming test on eye drops medical devices? The test allows to verify the mechanism of action of medical devices, meet and responds to regulatory needs, and it is accepted by the notified bodies and is completely customizable. Furthermore, considering the multiple endpoint approach, which refers to the use of more than one outcome measure in a preclinical study to comprehensively evaluate the efficacy or the safety or the biological activity of a treatment, the film forming test could be combined with other endpoints or assays, such as absorption study or moisturizing test or antioxidant stress preclinical efficacy test. Moving on the application fields, the film form assay can be applied to all reconstructed tissues, including, for example, vaginal epithelium, oral epithelium, bladder and airway epithelium, and many, many other. But I'd like to underline that given the significant demand for gastroprotective testing, in recent years, VitroScreen has developed a specific film forming model to evaluating the barrier effect of gastroprotectors medical device on esophagus mucosa after inducing stress with simulated gastric fluid produced and developed in laboratory. How can I get more details? For further information about the test and our test services, or for example, validate models and those models under development, you can visit our website and our LinkedIn page, and you can also contact us at our email addresses. And to conclude, in brief, who are we? VitroScreen is a company with over 20 years of experience, dedicating to delivering solutions in all fields of in vitro testing, focusing on excellence and innovation. Since 2021, we have become part of a Centogen group, and then now the mission of the group, consisting of VitroScreen, Centogen, and ToxHub with Carla and his team, her team, is to deliver top-tier services in vitro safety, efficacy testing, complemented by expert regulatory support. Thank you very much for your attention.
Francesco, thank you very much for your presentation. It was a very deep insight inside the techniques and your skills on this topic, so thank you. And last but not least, we go on with the presentation of my colleague Valeria Mazzoni. So please, Valeria, now the floor is yours.
Thank you, Luca. Thank you, everyone, and welcome to the last part of this webinar, where I'll be guiding you through extractable and leachable in non-solid medical devices, highlighting the importance of a strong analytical strategy. So, you know, extractable and leachable studies are central to patient safety and regulatory approval. So our goal today is to define the main points of a risk-based approach of this kind of project, aligned to the ISO 10993 and the USP guidance, beginning with understanding the materials, selecting worst case extraction conditions, and using the right analytical tools. So liquid and semi-solid devices in particular bring unique challenges, and we see together who they are and how to manage them. Finally, we'll see how a successful extractable and leachable program means coordinating different factors from chemistry to toxicology to ensure a robust, defensible, and regulatory ready project. So starting from the beginning, what are extractable and leachables? Extractables are chemical compounds that can be drawn out of the materials when it's exposed to artificial aggressive conditions, for example, high heat, solvents, or extreme pH. These conditions don't necessarily reflect normal use, but they are designed to stress the material and reveal what could potentially come out. Leachables, on the other hand, are the compounds that actually migrate into the final product or into a patient under normal real-world conditions of use. And these are the compounds that regulators are especially concerned about because they pose a direct exposure risk. Extractable and leachable studies are more than analytical exercises. They are fundamental to demonstrating and meeting regulatory expectation. And to evaluate any medical device, especially those in contact with the liquids, we must first identify all compounds that could migrate from materials into the patient pathway. And without understanding what these chemicals are, we cannot reasonably assess their safety. Next, we need accurate quantification. Regulators expect us to establish not only what is present, but at what level, because even well-known compounds can pose a risk when exposure exceeds toxicological thresholds. So quantification provides the bridge between the chemistry and the patient exposure. And finally, we perform the toxicological risk assessment, translating chemical data into meaningful safety conclusions. When these elements, so identification, quantification, and toxicological evaluation work together, they form the evidence base required to demonstrate biocompatibility. And this integrated approach is exactly what regulators expect. So a scientifically justified end-to-end assessment that ensures the device does not introduce unacceptable risk. In short, strong extractable and leachable science is essential to achieving both patient safety and regulatory approval. Optimizing extractable and leachable studies begins with a deep understanding of the materials and the intended use of the medical device. So by knowing the polymers, the additives, manufacturing processes, and the patient contact conditions, we can design extractable and leachable studies that are both scientifically justified and truly reflective of the clinical reality. And the second pillar is the extractable study itself, where we apply controlled, so exaggerated or exhaustive conditions according to both ISO 10993 part 18 and 12 to reveal the full chemical profile of what the materials are capable of releasing. And this type defines the worst case scenario and guides method development for targeted leachable testing that represent the third pillar. The leachable study is performed under simulated or real-time use conditions. Here, we can measure the actual compounds that migrate into the product or the patient pathway, generating exposure-relevant data that regulators rely on. And with no less importance, we must consider the correlation of the extractable and leachable results within the biological characterization. In fact, extractable and leachable data must align with cytotoxicity, sensitization, irritation, and systemic toxicity outcomes to demonstrate that any chemical exposure does not compromise biocompatibility. And this integration ensures our conclusions that has to be comprehensive, consistent, and defensible, of course. So optimizing this kind of studies requires marrying material science, analytical chemistry, and biological evaluation to deliver a complete risk-based safety profile for the device. But when designing extractable and leachable studies, one of our most important responsibilities is to use stressed extraction conditions that challenge the system, yet do not alter the fundamental state of the formulation or of the material. So stress conditions such as elevated temperature, extended contact time, or aggressive solvents are essential to reveal the chemical species a device could release under worst case scenarios. scenarios. But if those conditions degrade the formulation or damage the material in ways that would never occur during the real use, the study no longer reflects true patient risk. So when we stress the system excessively, we move from this to this, and it's not good. So our goal is to strike the right balance and conditions strong enough to fully uncover extractable, yet controlled enough to avoid including artificial degradation, polymer breakdown, or formulation instability. And this requires a deep understanding of material chemistry, product design, and clinical use too. And when extraction conditions are scientifically justified and non-destructive, the results become meaningful. And they allow us to properly identify and quantify compounds and then correlate that data with toxicology and biocompatibility assessments. In particular, liquid medical device presents some of the most demanding challenges for extractable eligible evaluation. First, we often work with very small sample volumes, which limits the amounts of material available for analysis and requires highly sensitive, low background methods. At the same time, these products typically contain extremely low concentration leachables, pushing our analytical system to their detection limits and increasing the need of clean workflows and robust quantification strategies. And the complexity of the formulation matrix adds another layer of difficulty. So, for example, surfactants, buffers, oils, and biologics can mask or interfere with the detection of the leachables, requiring selective extraction, separation, and of course, orthogonal technique to obtain reliable data. And one of the greatest analytical challenges remain the identification of unknown compounds. So considering low-level signals and matrix interferences, high-resolution mass spectrometry, fragmentation analysis, and expert review, of course, become essential to build confident structural assignments. And finally, all findings must be interpreted through scientifically justified threshold settings to determine which compounds require a full toxicological evaluation. And setting these thresholds correctly is a critical point to balance sensitivity, relevance, and regulatory expectations. But in general, how can we design an effective extractable eligible studies on these medical devices? So optimizing the studies begins with designing extraction conditions that are both scientifically justified and non-destructive, as I said before. So we carefully select solvents, temperature, and contact times that challenge the material enough to reveal potential extractable, yet do not artificially degrade the device or alter the formulation. And this balance is essential to generate meaningful worst-case data. Once extraction is optimized, we move to detection using a combination of screening and targeted methods. And the final step is the identification and quantification that transforms analytical signals into actionable safety information. So together, all these elements, so optimized extraction, robust detections, and precise identification and quantification build a strong scientific foundation for the assessment. And this integrated approach ensures that medical devices meet regulatory expectation and, most importantly, remain safe for patients. But let's keep an eye on each of these main bullet points, starting from the extraction optimization. So please go on, Dana. Okay, so extraction optimization is essential to generating meaningful and defensible extractable data. And to do that, we have to carefully design it. So starting from extraction conditions, they can challenge the material without causing unrealistic degradation. And to do that, we can use simulated use condition, exaggerated conditions, or excessive condition, depending from the contact of the medical device. As a second point, we have to select the solvent. This can be a critical point because they must represent worst case polarity while still being compatible with the material and the analytical methods too. And for the reason, at least the use of 2 solvents of different polarity is needed. And specific solvents will be needed also if the test aims to correlate chemical data to the results of biological testing. And finally, equally important are the extraction temperature and time, which must be elevated enough to accelerate the release of potential extractables, yet controlled to avoid altering the formulation or damaging the device. And by balancing these factors, conditions, solvent strength, temperature, and exposure duration, we ensure that our extraction studies reveal the true chemical risk while staying scientifically justified and relevant to clinical use. So moving to the second point, so to the detection. Effective detection relies on combining screening and targeted analytical approaches to build a complete and reliable chemical profile. So screening is our broad exploratory step using high-resolution full scan technique to capture as many chemical signals as possible, including both unexpected or unknown compounds. And it allows us to see the full landscape of what might be present without assumption or predefined targets. And to do that, a multi-determinative analysis involves various techniques is essential in order to detect different families of compounds, such as volatile compounds, semi-volatile, and non-volatile organic compounds, detective respectively with GC mass and LC mass, through an untargeted approach. But screening alone is not enough. In addition, we have to work with a targeted approach, for example, for elemental impurities by ECP, MS, and anions by ion chromatography. And together, screening and targeted detection create a powerful workflow. In fact, this dual approach provides a robust foundation for identification and toxicological assessment. Finally, moving to the last point, so the identification and quantification. These are the core of extractable leachable evaluation for medical devices, and they are often the most challenging steps. Small sample volumes, low-level chemicals, and complex matrices require high-resolution mass spectrometry, advanced separation technique, and expert interpretation to confidently identify both known and unknown compounds. And once identified, quantification is essential to assess real patients' exposure. And we calculate concentration and compare them to toxicological limits using the analytical evaluation thresholds, which are derived from the dose-based threshold divided by an uncertainty factor, calculated considering the relative standard deviation of the response factors from reference databases to account for analytical and biological variability. And this ensures that we focus on compounds present at levels of potential concern while filtering out all negligible signals. Together, so identification, quantification, the use of specific analytical evaluation threshold provide a robust framework to assess safety. And they allow us to determine what patient could be exposed to, at what levels, and whether those levels are within acceptable toxicological limits. So in extractable leachable evaluation, combining qualitative and quantitative analysis with orthogonal analytical technique is essential to building a reliable and safety profile. That's where orthogonal technique come into play. By using multiple complementary analytical methods can confirm our findings, improve sensitivity for different chemical classes, and mitigate the risk of false positive or negative too. And these layer strategies ensure that our extractable and leachable conclusions are comprehensive and regulatory ready, giving us confidence that we've captured everything that matter for the patient safety. And as seen, optimizing this kind of study begins with a well-designed extractable study. And using controlled condition, we reveal the full chemical profile the materials are capable of releasing under worst-case scenarios. This provides a comprehensive map of all potential compounds. And these findings guide the leachable studies, which focuses on compounds that actually migrate into the product or the patients under a real-life simulated use condition. And each device or formulation is assessed by a case-by-case consideration, considering all the factors like the storage, the administration, and the patient exposure. Exactly as for the extractables, qualitative and quantitative screening is necessary to identify all possible leachables. And with the same mindset, we can define that orthogonal technique provide confirmation across different chemical classes, ensuring robustness and strengthening the reliability of the data. So, combining together all this information, we reach the final step of the study. So please go on, Dana. And combining this, we can move to the draft of the final report, in which details of the entire study are summarized, including test article preparations, the description of any dilution concentration and other significant processing step form, and description of the analytical methods used, the IT calculation, and all details on qualitative and quantitative analysis. So, in conclusion, extractable and leachable testing is a critical point of the entire evaluation of the medical device, but it's fundamental to ensure its safety, efficacy, and regulatory compliance. And everything is built around the importance of the patient safety. So both chemical and biological characterization plus packaging or material compatibility become essential, and the systemic risk-based approach is crucial to minimize the risk from material selection to the toxicological assessment. And as final take-home message, I want to focus on the importance of an early extractable and leachable evaluation. So as said before, we have different challenges to manage, law and timing, contact conditions, the necessity of a straight correlation between numerous aspects that can keep all together to an extreme extension of this project. So an early integration could potentially reduce late-stage criticism. So thank you for your attention.
Thank you, Valeria, for your presentation, for the concept. And sorry, everyone, we are a little bit in delay today, but many different topics, full and dense of concept and knowledge, and we take some more minutes. So I think that now we can go through the question and answer session. Just to organize this part, we have just received some questions during the registration of the participants. So I think that we have time to go through these questions, and then eventually any other question, please feel free to write it in the chat and the question and answer section. And we will, as I wrote to you in the chat, answer to these questions after the webinar and send it back into the next days. So I think that we go through the question that we have just received, as I said. So the first one is, how do you determine the right balance between applying sufficient stress extraction condition without degrading the material or formulation? It's for you, Valeria.
Yeah, it's for me. So it's quite fundamental determining the right balance for stress extraction condition, as said during the presentation. So it's quite important to begin by evaluating the chemistry of the materials or of the formulation to understand how they respond to solvents, temperature, and extended exposure. And this allows us to choose the right extraction condition that has to be aggressive enough to reveal worst case extractables, while still avoiding all conditions that will chemically degrade the device or alter the formulation in unrealistic ways. So one possible route is running small scale feasibility studies to ensure that the system remains intact. And throughout the process, we rely on orthogonal analytical technique to distinguish true extractable from all the artifacts that can be caused by overstressing the material.
Thank you very much, Valeria. So we can go through the second question. Is the film forming test a quantitative method? I think it's for you, Francesco.
This question is for me, Luca. Thank you very much. Yes, sure, the test method is quantitative and it's based on the quantification of caffeine passed through the epithelium using HPLC techniques. Both the test and the analytical method were validated internally and by our external laboratory, giving us solidity to the method and, of course, to the results that we will be obtaining during the test.
Thank you very much, Francesco. So we can go through the third question. How do you ensure that the biological evaluation plan is complete and aligned with MDR expectations? So I think this is for you, Carla.
I think so. Thank you. The PEP, the BP, as I told you, is very, very important, is a fundamental part of the biological evaluation assessment. We start with by analyzing the device type, the formulation, the packaging, and the intended use. Then we identify the relevant biological endpoints, and we define the strategy to evaluate absorption, toxicity for both ingredients and extractable and leachables, and extractable and leachable analysis. That is a very important part of the biological evaluation. So this assessment help us anticipate the safety issues, also to avoid unnecessary testing, mainly for legacy devices. So yeah, a well-designed BP ensure that the biological evaluation report will be complete, defensible, and fully aligned with the ISO 10993 and the MDR regulatory requirements.
Thank you very much, Carla. So going quickly to the next one. Okay, is film forming a validated test? Just returning to question 2, I think this is for you, Francesco.
Yes, it's for me, Luca. Thank you very much. Yes, the test was validated internally, as I mentioned before, and the validation method was performed on negative and positive control, the Vaseline in question, with a number of biological replicates of 3. The aim of the validation was to define and the acceptable criteria of positive control, and in this case for Vaseline, produced with well-known film forming efficacy, and which represents the positive control in our experimental model. So basically, the experimental test data met the acceptable criteria defined through our historical analysis of previous studies, and the Vaseline should not show a caffeine passage greater than 40%, with a standard deviation between biological replicates less than 20%. That's the overview of validated the validation method performed in VitroScreen.
Thank you, Francesco. Thank you very much. So next one. Okay, so what criteria do you use to decide the appropriate real-life simulation condition for leachable study on a case-by-case basis? Valeria, this is for you.
Yeah. So when designing leachable studies, selecting the right real-life simulation condition is always a case-by-case decision, grounded in how the product is truly used. So we start by examining the clinical scenario, for example, how long the liquid is in contact with the device, how it's administered, and the patient's potential exposure. So we then consider the formulation, so the pH, the solvent characteristics, the viscosity, and the presence of any surfactants or biologic that could influence the leaching behavior. So the materials of the container closure systems that can also guide us. So considering the polymers, the coatings, or any adhesives that may interact uniquely with the formulation. Moreover, the storage or the transport conditions must also be reflected in the study. So altogether, this information can be useful, very useful to build a robust extractable leachable design.
Thank you, Valeria. So we go through the next one. Is performing an in vitro corneal permeation test enough, or its own to rule out systemic absorption? I think this question
maybe it's for me or
Carla.
for Francesco.
Yeah.
For me. Okay. And
yes, this is for you, Carla.
Yeah, okay, thank you. First, I'd say that we have to follow a case-by-case approach. I'd say not by itself, in vitro corneal permeability models like 3D models, reconstructed corneal epithelium, are very useful, but they represent only one of the possible absorption routes. Substances can also be absorbed through the other different tissues. So I'd say no, but again, we have to follow a case-by-case approach.
Okay, thank you very much. So the last is how do you approach the identification and risk assessment of unknown leachable when they are present at trace level in complex liquids formulation?
So I take
Please,
it.
Valeria.
As said before, the identification and assessing unknown leachables at trace levels, especially in complex formulation, is one of the most challenging aspects of extractable leachable studies. The first hurdle is the analytical part, and these compounds may be present below the typical detection limits, masked by also the formulation metrics, or can be structurally unknown. So to tackle this, we have to rely on the high resolution mass spectrometry, for example, fragmentation analysis, but most of all, the use of orthogonal technique is essential to confidently assign structures, even for compounds present at extremely low concentration. So once identified, our important collaboration with partners like ToxHub, for example, lead us to evaluate the toxicological challenge. So how to evaluate the risk when the compound is new and exposure is minimal. And here the become very important, crucial, the threshold. So the evaluation of the IET, the analytical evaluation threshold, that has to guide us in prioritizing which compounds need further toxicological assessment.
Thank you very much, Valeria. So we are 12 minutes out of our time, but I think it was very important to go through to this question that we received from all of you. So as we are at the conclusion of our program, we'll send out an email in the coming days, as I was saying at the beginning of today's journey, containing the survey form, the recording of the webinar, the slides, and the question and answer document. Additionally, if you want to have more news and updates from Neotron and Cotecna, you can subscribe our newsletter by using the link that we are going to send to you. So I hope that this webinar was beneficial to all of you, and a big, big thank to our speakers. So Carla Landolfi from ToxHub, Valeria Mazzoni from Neotron, and Shirley Francesco Carriero from VitroScreen. Thanks to all the colleagues for sharing the expertise, and to all of you for joining us today. So have a great day, and see you in the next webinars. Thank you.